One of the things we're going to do at Thwaite Gardens is a water quality survey. 1 in 8 people on the planet don't have access to clean water, so rely on freshwater sources such as lakes and ponds for their water, and these might be heavily contaminated with either chemical contaminants or biological contaminants. So water surveying is really important for environmental health. So we're going to use the gardens as a case study for doing water quality analysis. So when we get to the gardens, you're going to identify 2 different field sites to take samples from. You'll need to think about your safety when you choose those field sites, and also think about areas that have differences in water quality. You'll want to make some observations about those sites. You might want to take some photographs of them so that you really know where you've taken your data from. We're then going to analyse several different aspects of water quality. So first of all, we're going to look at various different physical and chemical parameters. So we're going to look at the colour of the water, so any changes in colour might be as a result of contamination. We're going to look at turbidity, which is the cloudiness, that tells us things like how much silt is in the water. We're going to look at the pH of the water, so is it acidic or alkaline. We're going to look at the conductivity, so that's how well the water can conduct electricity, which indicates how many ions are in the water. And we're also going to look at the oxidation reduction potential, so that tells us the oxidising power of the water. So, for example, if you have chlorine contamination, then that can affect your oxidation reduction potential. To measure the colour and the turbidity, we're going to be using a photometer. So these work on a fairly simple principle of you compare the amount of light going into a sample with the light going out of a sample, and that can tell you about what's happening in your sample. For the photometer, we need to set up what's called a blank, so that's a reference sample, so that effectively sets 0 on the machine, and then we compare it with a sample. So when we measure colour, we're going to use distilled water as our blank, and then a filtered sample of lake water in order to see if there's any change in colour. When we look at turbidity, our blank will be the filtered sample, and then we'll compare that with the unfiltered sample to see how much particulate matter there is in the sample. To look at pH, conductivity and oxidation reduction potential, we're going to use an ultrameter. So these are handheld meters that can measure various different things. You'll need to calibrate those meters when we go down to the gardens. We'll tell you how to do this, but the basic principle is you need to expose the meter to known solutions. So we have 442 is a standard that is representative of fresh water, so that will have the right ionic concentration for calibrating conductivity. We'll also have some pH calibration solutions, so pH 7, 4 and 10, so that will calibrate the pH, and in doing so it automatically calibrates the oxygen reduction potential. So biological health is also really important when looking at water quality. So we're going to look for the microbes that are in the water. So we're going to use a dipstick test. So you basically put your dipstick in the water, and then you put we'll collect in the dipsticks, and we'll put them in an incubator overnight for you. So that will allow any microbes in the lake water to grow up. So it allows us to tell us how many microbes there are in the water. So that could be an indication of, for example, contamination with sewage. And then we're also going to do an analysis of the invertebrates in the water. So looking at the creatures that are in your water sample is really important, because some creatures are more sensitive to water than others. So, for example, water snails are pretty tolerant to pollution, so they're quite common. So we'll give those quite a low score in our analysis, whereas things like mayfly larvae or dragonfly larvae are much, much more sensitive to pollution. So if we find those in our sample, then that's an indication that the quality of the water is quite high. So by looking at these different aspects of water quality, remember we've got physical, chemical characteristics and biological characteristics at 2 different sites within Thwaite Gardens, we'll get an idea of the quality of that water. Remember, while you're working, you'll need to take detailed notes of everything you're doing. You'll need to divide up the tasks between you and your group to get everything done within the time, and do be careful about your safety when you're working near the lake. We'll be giving you life jackets and waders, but please do think about your safety as you're working.